Hello, everyone, and thank you for taking a moment to learn about our research. Now, the project that I'm going to describe to you really builds on years of work in our laboratory where we have focused on Duchenne muscular dystrophy. And in particular, we have been developing new mouse models that carry the same types of genetic duplications that can be seen in individuals with Duchenne muscular dystrophy, and we are exploring how CRISPR gene editing can be used to correct those errors. Now, what makes this study especially important is that for the first time, really, we are looking at how CRISPR gene editing works when it's combined with a newly approved anti-inflammatory drug called vamorolone. More and more individuals living with Duchenne muscular dystrophy are taking medications like vamorolone to help reduce inflammation that then in turn protects their muscles. So a key question we need to answer is, how will these drugs interact with future CRISPR treatments? Will they make gene editing work better? Will they actually reduce its effectiveness, or will they have no impact at all? Now, these are really important clinical questions as we think about how can we safely bring gene editing therapies to patients in real life. So in the first year of this project, we created a new mouse model that carries a duplication of exon 44 that's directly based on a patient with Duchenne muscular dystrophy. What's especially exciting is that these mice don't just carry a mouse version of the gene, but instead, they carry the entire human version of the dystrophin gene, which means that we can test the exact same CRISPR therapy in mice that hopefully one day we would use in patients. And during that first year, we also developed and tested a CRISPR strategy using muscle cells from that patient with this duplication, and we were quite excited to see that we could successfully correct the genetic duplication. During the second year, we carefully studied how severe the disease was in these mice because we needed an important baseline that we could compare against later. As expected, and we were relieved to kind of see this, these mice showed classic signs of muscular dystrophy with reduced muscle strength, increased inflammation, increased muscle fibrosis, and so on. We also completed the manufacturing of the combined CRISPR and vamorolone treatment during the same time. So now we are in the part of the third year, and we just literally, as we speak, have started the treatment of our first group of mice. And over the summer, we will be evaluating their muscle strength and overall function, where we really then just compare three groups, right? So we'll have one group of untreated mice. We will have mice that are treated just with CRISPR, and then mice that are treated with CRISPR and vamorolone. We anticipate that the combination approach, or at least that's our hypothesis, will provide the greatest improvement in muscle health. And we're excited to do these analyses very soon, so keep your fingers crossed, please. Ultimately, this study is about more than just one experiment, though. It's really about understanding how gene editing therapies may potentially interact with medications that many, if not most, of Duchenne muscular dystrophy patients are already relying on. And that knowledge is actually quite essential to ensure that future CRISPR treatments are not only effective, but also safe as we move closer to clinical use. Thank you for your interest in our work, and most importantly, thank you for supporting our research. We couldn't do it without you, as we are trying to improve the lives with people living with Duchenne muscular dystrophy. Thank you.